Alright, hey, my name's Mario, and this is APES, period 6, and we are monitoring the conditions of the tank, and if you point at the, it's 5.4 milligrams, we can see that there's a probe, and that's a probe, and then there's a stirring thing, and that's ice water around, and in the middle you see there's a cup or a glass, and yeah, so that's our data studio. Yeah, and all right, so.
And today in this lab, we are going to see what factors can affect the rate of oxygen production in photosynthesis. And this is our lab. Photosynthesis is when carbon dioxide and water plus sunlight.
That's the sunlight.
That's the sunlight.
Your sunlight.
React to create oxygen, which is what we need and other animals need to live.
And plants. And you can see the plant, it's inside right there. Yeah. Okay. Alright, so we got some predictions before the lab. It was our hypothesis. That we thought it would decrease by I mean a significant amount. Okay. And when we did it, we kind of got a little bit of a different answer. These were the results. It did decrease a bit, but I mean it pretty much stayed the same. It started at 5.4 and it decreased to 5.2. The red line, it represents the first run, which was without the plant, and then the blue line, it started decreasing like after we put the plant in and the light, you know. And this occurred in 15 minutes. This was in 15 minutes.
Okay, so if we were to do this experiment again, I think we would like to know if there was carbon dioxide in the tube before the plant. In conclusion, we think that the photosynthesis didn't work as well because there was cold water in the tube, which was produced by the ice cubes.
So my prediction for this lab is that once we put the plant into the water, the oxygen level will rise because the plant will be carrying out photosynthesis. I think that the light will accelerate the production of oxygen, so the oxygen level will go up higher.
We found that when we put the plant into the water, that the dissolved oxygen levels almost stayed exactly the same, but it did have a slight decrease than without the plant in the water.
We think we got these results because there was no carbon dioxide present for the plant to carry out photosynthesis. Without carbon dioxide in the water, the plant wasn't able to produce glucose or oxygen, and that's why the dissolved oxygen in the water did not increase. Since plants also use oxygen, the dissolved oxygen in the water slightly decreased. Another experiment we could do to get better results and demonstrate the principle of photosynthesis would be to seal off a plant in a glass container with all of the essential nutrients it needs and carbon dioxide and sunlight. With the plant sealed off, we could leave it for several days and then measure the oxygen inside of the sealed container with an oxygen sensor. If the oxygen goes up, if the oxygen level goes up, we'll be able to see that the plant did produce oxygen and was able to complete photosynthesis.
We learned that without all three reactions, photosynthesis is not possible.
Hello, my name is Stin.
Stin! Go.
Hello, my name is Doug.
Doug!
And I'm Joe.
Joe!
The purpose of this lab is to notice the changes in the release of oxygen from a controlled group of a simple cup of water to a group with a plant that has a pre-existing amount of oxygen. As the light shines upon the plant, it synthesizes the effect of photosynthesis, which in effect releases oxygen from the plant, which would change the amount of oxygen in the total experiment.
The equipment for this experiment required a Mac computer with Data Studio, a GLX, a Pasco photosynthesis tank, and there was also a large rubber stopper, a temperature sensor, a dissolved oxygen sensor, as well as an Elodia plant.
The setup is the Pasco photosynthesis tank with water and ice in it, with a glass inside of it containing the more water and the aquatic plants. The lamp acts as a fake sun and makes the plant give off oxygen. The temperature meter just lets us visualize the temperature on the screen, and the oxygen meter lets us see what's going on with the dissolved oxygen. And the GLX is pretty much telling us everything, and then it's all displayed on the map. Simple as that. Alright, we're done.
The process of photosynthesis.
For the 8th grade minds.
The process is when light shines down on the plant, which absorbs the energy from the sun, and then creates oxygen in a compound of carbon, hydrogen, and oxygen.
Our predictions for the lab are that the light, when shown upon the plant, will act as a surrogate for the sun, which will create a process of photosynthesis on a much smaller scale.
Our results are the graph as follows. The blue line indicates the dissolved oxygen. It decreases as time goes on because the plant is releasing its oxygen into the environment. That's it.
Results occurred because the lamp shined down on the plant, which caused the plant to absorb the energy of the light, thus having to release oxygen.
Go.
An alternative for this experiment could be to go outside on a nice and sunny day and use the real sunlight for the photosynthesis, except for a lamp.
Also, we could use something into a much larger scale to compare with the sun by using a much larger plant, such as the sun comparison with the light.
I'm really glad that we did this photosynthesis lab today. I really learned a lot. I didn't really know what photosynthesis was before this, but you know what? Now I've got a great background and foundation to learn more about it. Photosynthesis.